Do you do flow bead assays? Take a look at MPLEX and see how data analysis should be done. So you're doing flow-based bead assays and using 96 or 384 well plates. Great. And now you're going to use one of those cumbersome company-supplied software programs to analyze the data. Maybe there's an alternative. MPLEX is a fast, intelligent program that reads almost any FCS files and lets you analyze the data really fast. And you don't need dozens of these every time you want to analyze the data. Only one screen to see everything you need. It works with 96 well plates or 384 well plates. MPLEX has a unique plate map for both 96 well plates and 384 well plates, and this map is a digital representation of the entire assay. It allows one to define every well electronically, and the software is very intelligent as it allows one to link different wells and plot dose response curves directly. Data can be output to an NIH required format for archiving or standard CSV files. NIH requirements identify specific directories which contain the raw data, the resultant data, and the metadata. Or you can just output a stack of CSV files or raw data in any combination, or pretty much any way you want your data. And if you collect data from a cytometer that labels the wells properly, you can just read them directly into MPLEX automatically and fast, as few as one well, all the way up to 384 wells. So you want to gate those beads easily? Why play silly games? Just click Auto Gate. And this becomes this. Maybe you're using Luminex beads from a kit. No problem. You can map any bead from any kit easily, and we use the standard Luminex bead numbers: bovine, porcine, human, murine, whatever. Our clever software knows where the beads are based on a map you can enter and modify, and this map shows the Luminex bead numbers onto the scatter or the fluorescence plot. This layout map tells the software exactly where to locate the bead populations. Okay, so you have some idea on how MPLEX works. Let's look at more details. This box lets you see every well and gives you the number or percent of every bead. And here you see the results by concentration or intensity for every analyte. Here is the scatter or any other parameter profile including bead number for each analyte. And here are your standard curves. But if you want to click here, you can access rapid data output. Here you can plot any subset of data by just highlighting some wells. It has automated identification of samples based on investigator, subject, or replicate number. There's a stamp function to replicate almost any sample feature across or down, and data output to CSV or NIH required formats, and save functions, export functions, and print functions. MPLEX gives instant bead number, analyte ID, and population color in one box, or you can zoom in on scatter plots at will. You can also remove any well from the analysis and show the percentage of beads per well, or change from percentage to bead number per well instantly. So you can get an instant view of well concentrations, or median intensity plots, or just plot one analyte, or maybe two or three. You can select one analyte and gate that for scatter, bead number, or concentration, and you can drop any graphic into a PowerPoint or a Word file instantly. So let me show you in real time just how fast and clever MPLEX is. First, open the program by selecting the MPLEX icon. It only takes 1 second to open. You see the entire screen with all the components. The first thing in order to analyze a set of data is to select the folder containing the data, or go to the timeline and select a previously opened data set. As a default, the software will open based on any previously analyzed parameters from that data set. Next, select the assay by going to Options and selecting from the available assays. By selecting Edit, it opens that assay so you can check that it is correct or change it. Then you need to apply that assay to the data set by creating gates for every bead. This can usually be done automatically, so click AutoGate. All the gates are drawn and bead numbers identified. You can change these easily if the AutoGate function makes any mistakes. By drawing a box on the screen, you can highlight the bead areas, and if you again hit Edit, you can drag the assay window under your gates to check that the bead map correctly matches the AutoGated regions. You now have completed the analysis of your data. The only thing you need to do now is output the results. I hope that was not too time-consuming. The same process is followed for hormone data using Luminex type beads. Open the program by selecting the correct directory, and the software opens all the files associated with that directory and displays the last used analysis. If you zoom in on the beads, you can check that they are correctly associated. Now all you have to do is output the results. A key feature in MPLEX is the plate map. I'm going to show you quickly how this is created and used. The first thing you enter is the location of the standard curves. We suggest using 24 single curve points rather than the typical recommended 12 point in replicate. You must number these with a C followed by the dilution number. You can place them anywhere, but I suggest top and bottom rows. Next, enter the PI's name on the investigator box at the bottom. Here I'm using PI. If you highlight all the boxes in yellow and just type PI, they'll all be labelled, or you can drag the labelled boxes around if you like. Next, move to the subject line or line 2 at the bottom. Here we enter, for example, the rat number, in this case 083. And since we'll use one rat per line or row with 2 sets of 6 sample replicates, 083 will be on the entire row. Continue with each rat number as shown until the plate is populated. Next, select the replicate number called copy, on this case A for the first set and B for the second set, as we have now 6 rats and 6 samples for replicate A and 6 samples for replicate B. Next, all the first column samples will be time point 1, so highlight them and add 1 to the box at the bottom of the screen marked parameter. Highlight column 2 and enter 2 for all of that column and 3 for the next column, etc. for the entire plate. And we add a few odd samples at the bottom where we only have 4 time points. You can add comments to any well, and a red dot appears on that well to let you know that there's a comment. Once the plate is filled, associate this with the data set by hitting Apply on the top row. Now you can select the samples on the top right that link an investigator, a rat or a sample, and a selection. And it lets you see the results in the graph below. Alternatively, you can just highlight any wells and see the results in the graph. There are several ways to output data. One easy method is to just output to NIH required structure. At the top of the plate map are 2 buttons, CSV and NIH. Both create structured outputs. I'll demonstrate the NIH method here. When you select the NIH button, it asks you to select a saved directory, and when all the files have been formed, a message pops up saying MPLEX done. Now we can look at those files, and you see 4 directories: derivative, info, primary, and source. These contain all the data in CSV format and also include the original FCS files. There are also explanatory files that instruct the user in how to interpret the data and exactly how the data were run. Also, every animal will have its own CSV file with all the appropriate results.